Hello and welcome to Smith and Sheridan on Biotech, a podcast on the science and business of biotechnology, presented by me, Cormac Sheridan.
And me, Andy Smith. Hello, Cormac. Hello, everyone. How are you this week, Cormac?
I'm good, Andy. How are you?
I'm ebullient, as they say, because I've had a pretty good week. I've had my summer bike out, so I've done the fastest time on my summer bike this week, and I've got 2 personal bests on a short metric round in archery this week. But the fly in the ointment is that these are all activities that are done outside in summer. And that brings us to the subject of our discussion this week, doesn't it?
And I hope you took Baz Luhrmann's advice and...
Yes, I'm busily using up our house's supplies of sunscreen.
As been stated before, we do not provide any medical or investment advice on this podcast. Mm-hmm. But we can safely recommend the wearing of sunscreen at all times.
Or as the Australians say, slip, slap, slosh, isn't it? Is it?
Possibly. I have not had that conversation.
Slip, slap, slop, I think it is, yeah.
But melanoma obviously is a very serious form of skin cancer if it's caught late. The surgical resection is very successful if it's caught early and it's just localized to primary tumor. But it's a very, very nasty cancer if it has spread and metastasized. And I think we can also say, I actually think if you were to write a history of the biotech industry over the last 15 years, an awful lot of it would actually revolve around melanoma. It really has been an extraordinarily important proving ground for immunotherapy in particular, but also for small molecule targeted therapy. And it's an area where it's not a solved problem, but the outcomes are still streets ahead of where they were. I mean, in one commentary I was reading today, median survival, according to the study authors, had improved from about 6 months to 6 years in patients with advanced inoperable stage 4 disease. That's extraordinary. And the history, it's well documented. I actually think there's Jim Allison's 2018 Nobel Prize acceptance speech or lecture is a very, very good place to start if you want to get into the history of the stuff. The 2011 FDA approval for ipilimumab or Yervoy, the Bristol Myers Squibb drug, which inhibits CTLA-4, that kind of kick-started the whole immunotherapy movement, especially around checkpoint inhibition therapy, rather, across all cancers, really.
Yes, it did. But before that, I think we need to go back further than that. We were in a similar situation to we were discussing last week with brain cancers, where, the options years ago were only surgery, radiation, and very nasty chemotherapy.
Yep.
And then we have the biotech and the pharma sector rising to that challenge. And it makes you wonder why in malignant melanoma and not in malignant glioma. Well, partly it's because the money isn't there. Malignant glioblastoma is a very rare cancer. And by contrast, malignant melanoma is a very common cancer. I saw it's about 1/6 of the US common cancers, and it's not going to get any better, right? Because global warming, aging population, so more sun exposure cumulatively to people as they live older is going to give us more cases. So quite right. Hats off to the sector that has come with and is coming with all these new therapeutic interventions. And there is going to be a market there, right?
There is already a large market.
There is already a large market, yeah.
Yeah, and I must say I'm much more focused on the outcomes than I am on the financial rewards, because what we have is good outcomes for people with early stage disease. We have responses for many people who have more advanced disease, but unfortunately the responses oftentimes aren't of sufficient duration.
And there seems to be a sort of cut-off. And as you were describing earlier, this is one of the few cancers. And again, contrasting brain cancer we talked about last year, this is largely a wholly preventable cancer, right? Don't go out, don't expose you to sun, slap yourself full of sun cream.
And wear protective clothing, how about that?
Wear protective clothing, yeah, and you reduce the incidence. And it's one of those few cases where you can prevent a cancer. But then I was reading, as you described, the earlier stages, if you find it early enough, a biopsy that goes 5 millimeters down into the skin that can actually remove that whole tumour, and then you won't have melanoma after that.
Yeah.
But what we're spending much of our time on is the treatments, and these are the patients where you don't know, you don't see it until you're too late, or you don't ever see it at all. If it's on the top of your head or the back of your neck or something, and no one sees it, and then that metastasizes, and it can go anywhere, and it's nastily aggressive when it does.
Yeah, it is. And to focus on the sort of the biotech industry aspects of it rather than the hardcore medical aspects of it, where you and I obviously don't have a whole lot of expertise or any, rather. It's fascinating to me, though, that the evolution. I mean, if you look back from that Yervoy approval in 2011 and where the industry has edged along since. I mean, back then, the pivotal trial from which Yervoy gained its approval, basically, there were co-administering it actually with the GP100 vaccine, but the vaccine itself didn't seem to be active at all. So whether they were on Yervoy or on the combination of Yervoy and the vaccine, they had a 10-month median overall survival versus 6-month median overall survival for those on the vaccine only.
I mean, that's good for those patients who get those extra few months, right?
It is, yeah.
I remember at the time, I think the industry and even the press was particularly nonplussed at the approval. And it's fair to, I made the mistake some time ago in a podcast, and you actually agree with me, Cormac, when I said sometimes the first drug approved is the best one for that indication. And ever since, we've had examples where that's not the case. And, CTLA-4 inhibition for melanoma has proved one of those, because it never really got going until the PD-1s started happening, did they?
That's correct. But then when you did a combo, and BMS obviously deserves huge credit here, they bought Medirex on the back of the ipilimumab data when it was still undergoing clinical trials. But they chewed away at it and they added the PD-1 inhibitors. So they went, and again, now this is interesting. And again, it's a different trial, right? So you can't necessarily compare and contrast. But when they did a pivotal trial of their IPI and Opdivo or nivolumab, their PD-1 inhibitor, the median progression-free survival in the pivotal trial there was 11.5 months for the combo regimen versus 6.9 or 7 months for nivolumab alone, or 2.9 months for ipilimumab alone. This is the CheckMate 067 study. Very well conducted clinical trial. In the combo group, at minimum 60-month follow-up, the median overall survival was not reached for those on the combination. For those on nivolumab, it was 36.9 months, and for ipilimumab, it was 19.9 months. So that really, to me, is the sort of high watermark of the PD-1/IP story, or the immune checkpoint, until we get, and again, Bristol Myers Squibb, to their credit, they've followed up with another immune checkpoint inhibitor combination. This is the first instance of a LAG-3 inhibitor getting an approval. Yeah. So they have a brand name for the combination of the two antibodies, Opdualag, which is relatlimab plus nivolumab, the LAG-3 inhibitor plus the PD-1 inhibitor. And in the phase 3 clinical trial that gained the FDA approval here in 2022, the median progression-free survival for the combo was 10.1 months versus 4.6 months for the nivolumab alone. Again, the overall survival was not reached in the combination arm versus 34.1 months nivolumab only arm. So what's key here, though, is that they are really pushing hard on getting sort of equivalent efficacy for the LAG-3 inhibitor and PD-1 inhibitor that you already get with the PD-1 and the CTLA-4 inhibitors.
Yes.
But you're getting it with the far, far easier side effects burden. I think that there's about sort of 1/3 less kind of serious treatment effects, 21% versus 59%.
And I think there are 2 points that interest me here, Cormac. The first is melanoma, or the use of the PD-1s in melanoma, had revolutionized the survival for those patients, because it's such a big indication. But it opened the door to so many other solid tumours. I mean, you think of, not just fourth line, but we're going all the way down to first line lung cancer at the moment. So the PD-1s were enabled, if you like, by melanoma, or given that chance, and they excelled in that.
Yeah.
And then we're off to the races on combinations. The other point I wanted to ask you about is, in terms of the temporal perspective, you think of CTLA-4 plus PD-1 or PD-L1s, And then, as you said, the combinations with lag. But then didn't the BRAF kinase and MAP kinase inhibitors come in between that point?
They were coming around the same time. Again, the BRAF inhibitor, well, it was a mutant BRAF inhibitor, Zelboraf or Vemurafenib. Plexicon, or remember Plexicon?
Yeah.
Now part of Daiichi Sankyo. Themselves and Roche were the ones who got that one over the line. And again, back in 2011, this is the sort of the stone age of melanoma therapy. In the pivotal trial there, treatment-naive patients with BRAF V600E mutation, again, they were unresectable or metastatic. Basically, they had a PFS improvement of 5.3 months versus 1.6 months. Overall response rate, huge difference, 48.4% versus 5.5%. But basically, 23% of the patients on the active drug treatment died during the study versus 36% on the comparator drug chemotherapy agent called dacarbazine. But fast forward, that was 2011, right? So they got that approval then. Fast forward then a couple of years later, just 2015, 4 years later, Roche again, they combined Zelboraf with another agent, Ortelic, or cobimetinib.
Oh, yes.
And that really jacked up the efficacy. They got median follow-up of 14.2 months. The median PFS for the combo was 12.3 months versus 7.2 months for Zelboraf only. And the median overall survival was 22.3 months for the combo versus 17.4 months for Vemurafenib only. So again, they just showed again, therefore, that combination agents, which, I mean, we obviously get the logic of it. Yeah. That was a MEK inhibitor, by the way, cobimetinib. So that was good work done in 2015. And obviously those drugs now, if they're not already on generic, they soon will be. And especially with people who have a BRAF mutation that renders the protein constitutionally active, that can be a very good treatment option either prior to or in addition to immune checkpoint inhibitor therapy. But then, what remains the problem, and again, say, Array Biopharma, another company named to conjure with, acquired by Pfizer back in 2019, they again had very good outcomes for another combination, another BRAF inhibitor and a MEK inhibitor, Braftovi or binimetinib, and Mektovi and Corafenib. A similar kind of outcomes, much superior progression-free survival for the combination arm. So that got an FDA approval in 2018. So then, though, whether you're on immune checkpoint inhibitor therapy or on small molecule kinase inhibitors, you generally reach the point that you become either refractory or you relapse. That's where things get a bit kind of difficult. They can try you on another combination therapy, maybe. They can fiddle around, obviously, with the regimen you're on. But it's the reason why there was a certain amount of excitement this year for the approval of the Iovance Biotherapeutics tumor infiltrating lymphocyte therapy, Amtagvi or lifileucel. And this really is aimed at patients who've either had a BRAF inhibitor and or a MEK inhibitor, or who've had PD-1 inhibition therapy. So these are patients
relapsed.
Yeah, and they don't have a whole lot of options. And they might have had, again, a much longer period of time where they remain alive that they wouldn't have been 10, 12 years ago. But the conditions come back and the doctors are wondering what to do. So this is Stephen Rosenberg from the National Cancer Institute, one of the great heroes of cancer immunotherapy. His work led to this. And when you think about it, it's an extraordinary approach. You get tumor-derived T cells from each patient. So you take a biopsy from a patient's tumor and you harvest the T cells from that and you give it a bit of a activation boost and you pop them back into the patient and you co-administer it with interleukin-2 to give it some further activation signal.
I think you need to amplify them as well, don't you, to get in?
Yeah, exactly. Yeah, you would certainly...
There must have been histopathologists for decades who've looked at tumours in cross-section and looked, oh, it's interesting. There's infiltration by T cells. And then perhaps, some immunohistochemistry that actually said, oh, they're activated T cells. But it's taken decades of that observation to come up with someone to actually try and design something that replicates that.
But what amazes me, though, about it all is the amount of science and precision that goes into this.
Absolutely.
Because this isn't something that's manufactured or cooked up in a lab according to a recipe or a manufacturing protocol. The quality control specifications and the release specifications define the product, as it were. But there's a huge amount of science that enables you to do this with a certain amount of consistency and therefore with certain expectations of results. Because, I mean, it is a bespoke therapy. So if I'm unfortunate to have metastatic melanoma and my neighbor down the street also has it, I mean, we're not getting the exact same therapy.
Yeah.
We are getting the same treatment and it goes through the same process, but there may be reasons why one person's activated T cells are more active than another person's.
Which is going to give the regulators some headaches in the future, isn't it? Because it's not the same product.
But they've approved it. And I mean, you could say the same for any autologous.
Well, you could say for CAR-Ts, couldn't you, as well?
Yeah, you could, yeah. But it's all about the specification and the assays that allow you to define the product. But again, what's very telling, though, if you look at the FDA label here, I mean, the kind of objective response analysis and all the rest of it was calculated on the basis of 73 patients included in the analysis. But 111 patients who were recruited underwent tumor resection, and 82 patients actually underwent manufacturing, but 9 of these did not get the required dose, which was the dose range was set at 7.5 by 10 to the power of 9 to 72 by 10 to the power of 9 viable cells. So 73 patients received Tagvi in that kind of dose range, and a bunch of other patients didn't. Now, you would hope in time that they could maybe refine and improve upon the protocols to reduce the number of manufacturing failures, because here it's over 10%. And if you include the people who were actually 111 who underwent tumor resection, now some may have died actually before they actually had the opportunity to do the cell extraction and the processing, but that's an issue. And again, the overall response rate here was 31.5%. But what's very interesting is that those who responded well, 12 months out after therapy, and it's just a one-shot therapy. But after 12 months of those who did get a response, over 54% were still responding. And I think that's quite amazing. So it might only be 15% or so, or just a little bit more than 15% of those who were included in the primary analysis. But that's, I think, something that, to me, it's always a question of the first therapy kind of prizes open the gap where the doors are firmly shut. If you get a signal at all, you then can work on it and prize it open further. And I think this is really interesting work.
But it does illustrate to me the power of the cellular therapies, be it TILs or CAR-Ts, because they persist, right? You give them to the patients and you don't have a half-life of a day or so where the drug elutes. These cells are then circulating around the body, going after cancer cells for up to a year or more afterwards. And
what's really amazing is that these patients had become refractory to PD-1 inhibitors, right? So that means that the inhibitory signal that's coming from PD-1 is active within their cancerous lesions. And yet, if you take out these cells and you give them a bit of a dare I say Viagra, that's very unscientific, but if you give them a bit of a boost, a shot of caffeine, as it were, they're back in business and they can do things within the tumor microenvironment that they were incapable of doing before this therapy was administered.
Oh, no, I definitely think Viagra's the right analogy, yeah.
Thank you. And then there's Obsidian Therapeutics is trying to come up with the next generation of a TIL therapy. They're doing this same thing, tumor infiltrating lymphocytes, but these are engineered ex vivo to express interleukin-15 and thereby allowing you to dispense with the need for concomitant IL-2 therapy, which causes some of the safety concerns.
Which has its own toxicities.
Yeah, precisely. So I was just able to find phase 1 data for this. And again, they had a very small number of patients, right? 9 patients, they had a 44% objective response rate, 50% in 6 patients who received dose levels of greater than 30 by 10 to the 9 T cells, tumor reduction seen in all patients. And so they're now doing a phase 1/2 trial. They're kind of obviously expanding on this initial small patient cohort. And I think it's very interesting. I think it's extremely interesting. And then there was a huge amount of hype around what Moderna and Merck are doing.
Which seem to be our old friends, right? I think this is the third time in a podcast we've mentioned them, but we mentioned them last week for brain cancer, and now we're talking about melanoma.
This is correct. This is correct. And I think it's the other side of the coin. Again, it's immunotherapy where you're trying to achieve lasting effects. And here, Moderna has come up with another neoantigen individualized cancer vaccine based obviously on its mRNA platform technology. And they're pairing it with Merck's Keytruda or pembrolizumab, best-selling PD-1 inhibitor in the industry. And they're doing a phase 3 trial, and they're comparing the combination with just the pembrolizumab alone. And in a phase 2b study, and these are patients with completely resected melanoma stage 3b or 4. So this really is, highly advanced metastatic disease. Yeah. The stuff where you really, in the past, had very few options. So they increased recurrence-free survival. I mean, the hazard ratio for recurrence of death was 0.561, which I think is a very, very impressive kind of number.
Yes. Yep.
So the recurrence of death event rate was 22% for those on the combination versus 40% for those on pembrolizumab alone. So that shows you that you're almost having, again, the risk of death or recurrence during the trial. And the 18-month recurrence-free survival rate, again, was 79% for the combination versus 62% for pembrolizumab. So pembrolizumab alone is a fine agent. Okay, it's better than ipilimumab, but adding the vaccine, you would hope that you just get greater durations. We need to see more data on this one, but I think it's very, very interesting.
For me, it helps the lineage or the chronology of the development of agents for melanoma. I mean, when I went to my first JP Morgan healthcare conference in San Francisco in 2001, you'd had biotech companies standing up, giving presentations, talking about their small molecule inhibitors of a particular cancer. And they would almost always use the phrase, no overlapping toxicities, because you worry about the cumulative toxicities, not just the chemotherapies that came before, then the small molecule inhibitors and whatever. And fast forward now, we have the interleukin toxicity, which people are trying to have interleukin sparing with melanoma activity in virtually the same way as, do you remember way back in hepatitis C, where you'd have ribavirin and you'd have a cytokine that would give an awful flu-like illness when they're trying to treat the hepatitis C? Well, certainly malignant melanoma patients have a higher tolerability for that. But with the small molecule MEK and other inhibitors, we seem to be having our cake and eating it, and we're having more active agents that are less cumulatively toxic. And that's a testament for the industry, isn't it?
Yeah, I think it is. They're plugging away at it all the time. And it is, I mean, I never really fully bought into your sort of the first drug in a class is often the best one.
Oh, you say that now.
I might have nodded passively to encourage you. But I often think, okay, it can be the case, but it can also be the case, though, that a first drug is a toehold or a foothold in an area. And then once you get that, you can prise it open. And I do think that immune checkpoint inhibitor therapy, they've had extreme difficulty trying to find the right combinations and trying to find the right dose regimens in which to deliver those combinations to improve the outcomes or to move the needle. It's been very slow, and the number of checkpoint targets that have come on are far, far less than I would have expected when I first started writing about this area 15 years ago. It surprises me greatly that we've barely moved beyond PD-1 and Opdualag and PD-L1. LAG-3, yes, finally, but only in this one indication and only in a combination setting. It's just been very, very slow to move because there are so many checks and balances involved in the activation of T cells and of other immune effector cells. And it's just been very, very slow to find the right levers and learn how to work them to get you the kind of outcomes that you want. And it shows to me that we don't fully yet have a handle on what's really happening within the tumor microenvironment.
But we do appreciate how aggressive this type of cancer is. And your lead into it at the beginning hinted that you can get an agent that addresses one particular molecular function of a tumor, and a very aggressive tumor cell will just laugh in the face of it and go round it metabolically or some other crosstalk in it, and they'll move because they grow so fast they can do without a lot of things. And For me, it just reinforces the fact that, the future of cancer chemotherapy and particularly malignant melanoma is going to be, there's no way about it. It's going to be in combinations. It's going to be in combinations of more and more advanced therapies. And we're still seeing that happening.
Yep. And it's interesting too, though, I mean, the sort of, I wouldn't say necessarily casualties, but those that have really failed to move the needle here in ways that we might have expected different outcomes. Imlygic is one product that...
Yes.
No, we didn't expect a whole lot of it even when it was in clinical trials, or TVEC, Amgen's modified herpes simplex 1 virus. That got FDA approval in 2015. And again, everybody thought this was going to be the start of oncolytic viruses.
Yeah.
It wasn't a great agent, but it was okay. Now, it was always safe and pretty harmless. Maybe that's why it got kind of over the line. But I mean, for those who are not aware of the history, it genetically modified live attenuated viruses, and it produces GM-CSF, granulocyte macrophage colony stimulating factor. A very, very, very long name for an inflammatory cytokine. And you could only do it in early stage disease, even though, no, it's actually, you were giving it to people with stage 3 or stage 4 disease, but you could only give it to them in lesions that were injectable, essentially, or biopsyable, essentially. So you couldn't give it to visceral metastases, but the hope was that you get a systemic immune response by administering the virus in certain of the lesions. And the median overall survival in the pivotal trial was barely above that of GM-CSF, 23.3 months versus 18.9 months. The numbers look kind of good, but it's not a very active agent. But again, I've never seen it paired with anything else that might move the dial further. It just hasn't really resonated with physicians. And to my great surprise, I've not seen anything else come behind it either. The oncolytic virus story, despite huge efforts, has yielded very, very little.
There are still companies working on it,
Yes.
some of them in the distant parts of Canada.
Yeah.
But as you say, I think its time has come. Oncolytic viruses for cancer have come and been de minimis and passed.
Yeah, I hate to write them off. I hate to write anything off, but sometimes you just need to work away at animal models and do the basic research. And sometimes you can maybe, I'm not saying this applies here, but I'm just saying that idea that there can be one finding in another area entirely that can open up new possibilities. I'd hate to write them off because the idea is such a clever idea. And the fact that they're easily programmable,
Yep.
generally can be quite safe.
Although the possibility exists, Cormac, that the first ever approved oncolytic virus for melanoma might just be the best, which is not saying much.
Yeah, I take a point because you were saying it's potentially a blind alley.
Yeah.
And it's interesting that Regeneron are in this field and they're just trying to, for once, doing from what I can see, a Me Too effort. They're trying to essentially do what BMS has already done with Opdualag. They have also a LAG-3 and a PD-1 inhibitor in phase 3. So it'll be interesting when those data come out.
And their PD-1, Regeneron's PD-1 inhibitor is already approved, right?
Yes, that's right. Libtayo, yeah.
Yeah, a bit of an also ran in the checkpoint inhibitor race, isn't it?
Yeah, but my sense always is that they just wanted to have their own PD-1 inhibitor so they could use that as a backbone in their own clinical trials.
Right.
It just gives them kind of freedom to operate, as it were.
Well, so did Merck KGaA, but it didn't really get them very far. I mean, I got them to Pfizer, but then it didn't get Pfizer very far either.
Yeah, Merck. Yeah, that's true. Yeah, and there's a few ones. BioNTech is in here, and actually that's the case in point. BioNTech has a fixed antigen vaccine, BNT111. So that's just encodes for melanoma-associated antigens. New York esophageal squamous carcinoma 1, or NYESO1. MAGE-A3 tyrosinase and a transmembrane phosphatase. So they've got an mRNA vaccine encoding 4 antigens associated with melanoma, and they're doing a phase 2 where they're pairing it with Libtayo or ipilimumab, Regeneron's PD-1 inhibitor. And, it's interesting that's still on their pipeline chart. They're still putting resources into this. It's in a phase 1 trial, 3 of 25 patients on the vaccine only had a partial response. 7 of 25 had stable disease. And then in a different cohort, 6 of 17 patients on the combination of 111 and the PD-1 inhibitor had a partial response. It's not setting the world on fire, but never write BioNTech off. And again, here's a very interesting one, though. This is a more interesting one. This is Evaxion Biotech of Copenhagen. They've just published data on this peptide vaccine, which is also directed against neoantigens. And they're using an AI antigen prediction platform, of course. So if it's AI, it has that sort of ding kind of little AI inside piece going on. But they attained an overall response rate of 67% in a small phase 1 study in patients who previously received either a PD-1 or PD-L1 inhibitor. That's interesting. So these are refractory patients or of 67%. So they're now doing a phase 2 in combination with pembrolizumab in patients with metastatic or unresectable disease. That's one to watch, I think. I think that's one to watch. But that's an exhaustive but not complete run through the options pipeline of melanoma therapies. There's more, and there's more to come. But I do think it's an area of biopharma, biotech, innovation, and there's been obviously a ton of investment has gone in to get us this far.
Yep.
And as we were saying earlier, it is not a solved problem, but at the same time, there has been very, very substantial progress in the last 15 years. So, without further ado, I would exhort you to continue wearing sunscreen.
Yes, and covering up.
And the other advice from that song, the Baz Luhrmann song, where it was to floss, which I think is also good advice, and also to dance. Now, I also think it would be important not to try these at the same time, because that could result in serious physical injury.
Oh, I don't know. I mean, there doesn't seem to be a lot of overlapping toxicities there.
Well, you could wear sunscreen while doing either the other two activities, but trying the three simultaneously is a recipe for disaster. Until next week, Andy.
Cheers, Encomic. Cheers, everyone. Bye-bye.